We are going to talk about complement today, specifically the classical pathway in complement. Now, this is often considered part of the adaptive immune response because it relies on antibodies. As you can see here in my drawing, I've shown you the cell membrane of your pathogen, something like a bacterial cell. And on the surface of that bacterial cell, there's antigens, and within those antigens, there's epitopes or short sequences of the antigen or the protein that antibodies can bind to. So because antibodies are required in the classical pathway, it's said to be considered adaptive immunity. But however, you'll see that many of the components produced in this part of the pathway or in this classical complement pathway are necessary and help boost the innate immune response. So it's sort of a bridge between both arms of the immune response. We're going to start this pathway off after we have our antibodies binding the antigens, as you see here. We're going to start it off with something called an initiator protein binding to the antibody complexes. Now, these antibodies that are first bound are IgG. You can have 2 of them, or you could have 5 IgM antibodies. None of the other subtypes are involved with classical. So for simplicity's sake here, I have 2 IgG antibodies that are bound to our antigens. And then we're going to see our initiator protein come in. This will be binding directly to the antibodies, so we'll draw these so it's interacting a little bit better. And this initiator protein is a large complex that's made up of 3 different types of complement proteins. The first one's called C1q, and then C1r, and C1s. Now there's actually 2 copies of C1r present, and there's 2 copies of C1s present. And when they all come together with C1q, we give it this very long name, so C1q R2 S2. Now this is our initiator complex that binds to the antibodies that are bound to antigen on the pathogen or our bacterial cell, and this is what gets the entire process started. So after that, what we're going to have happen now is there's going to be protease activity that's activated in this initiator complex. Specifically, the C1R protein that's part of the complex becomes active. And proteases, their job is to cleave other proteins. So what C1R is going to do is it's going to cleave C1s, which activates its protease activity. And you can start to see here now that we're starting to activate a cascade. So once this complex has protease activity activated now, the first thing that's going to happen in our cascade is a complement protein called C4 is going to be acted on. So here's C4, it's going to bind near C1q R2 S2 or initiator protein. And what's going to happen then is C4 is going to be broken or cleaved into 2 different subunits. We'll have C4b and C4a. Now when we talk about complement cascade, and we're going to see that a series of these proteins end up getting cleaved, usually the larger part that remains is designated B, the smaller part after the cleavage is designated A. One of the things about complement pathways is that through this series of cascades or cleavages that occur, we will generate a number of what we call inflammatory mediators. So we're going to store these up here in this part of the board as we go through our cascade. C4a happens to be our first inflammatory mediator that we generate. So we're going to just put that up there. What inflammatory mediators are is that these are small proteins that help increase the diameter of blood vessels. So they bind to endothelial cells lining the blood vessel walls, and they cause blood vessels to dilate. What that allows is for immune cells to infiltrate the site of infection, and it basically increases the amount of inflammation that's occurring during a response. This can be a good thing to fight off pathogens, but if you have too much inflammation, it can cause things like anaphylactic shock. So oftentimes you'll hear these inflammatory mediators, such as C4a, referred to as anaphylatoxins. Because in too high of a concentration, it can actually be detrimental to the person. but in a low enough concentration, it can help promote inflammation. So we have C4b that's been generated now from the first cleavage that occurred in our cascade. And what's going to happen is this is going to come down and bind near to the membrane. So we'll have C4b down here. Now, our initiator protein, C1q-R2-S2, still has serine protease activity, and the next protein that it's going to act upon is something called C2, or complement protein 2. So if we follow the same pattern as before, we're going to see that that gets broken down into C2b and C2a. In this case, C2b is actually the one that's going to diffuse away. It's no longer really needed, so we're just going to remove that from our board. And C2a is going to come down here and join up with C4b. Now this complex of these two proteins together forms an active enzyme, which we call C3 convertase. So C4b plus C2a is C3 convertase. You might also see it abbreviated C4b2a. And C3 convertase, you can probably guess what that's going to act upon. So it says it right there in its name. This enzyme is going to cleave a complement protein called C3. So if we follow the same pattern as before, the C3 convertase will act on C3. It will break it into C3b and C3a. Now, C3b is a very important molecule. The C3 convertase can actually make up to 200 C3b molecules per single C3 convertase. And so I'm going to put another C3b up here. It's because this is important in the innate immune response as well. What C3b can do is it can act as what we call an opsonin. Now, if you recall from previous lectures, opsonins are important in coating the surface of pathogens and providing a better recognition signal for phagocytes to see that a pathogen is present so that they can opsonize or engulf it and destroy that pathogen. So C3b, by having many molecules of that produced in the complement cascade, we're really increasing the opsonization process, or the ability of phagocytes to recognize pathogens and destroy them in the response. So now if we go back to our C3 being cleaved into C3b and C3a, the next thing that's going to happen is we're going to have C3a come over here into our inflammatory mediators section. So C3a is another anaphytoxin, similar to C4a. And then we're going to have C3b, some of those molecules come down here and bind to the proteins that made up our C3 convertase. So now we have a new protein called C4b2, B2a, 3b. Very long name, complement pathway gets pretty confusing, but scientists have abbreviated this 3-protein complex now as C5 convertase. So one molecule of C4b, one protein or subunit of C2a, and one of C3b, all together will make what we call C5 convertase. Sorry. So you can guess what that's going to act upon now. It's a C5 convertase, so this is an enzyme that's going to act upon our complement protein called C5. And that is going to break C5 into C5b and C5a. Now C5a is another one of those inflammatory mediators, so we'll move it over here into this section. It's an anaphytoxin. It's going to increase blood vessel permeability and help promote inflammation. C5b is actually a very important complement protein, which is going to come back down and embed itself near the membrane, or very close proximity of the membrane of the pathogen. And this is going to be responsible for setting up the next complex, which we call the membrane attack complex. This is very important in the classical complement pathway because it's a large complex which actually is going to poke holes in the membrane of the pathogen. So if C5b starts off the part of the pathway that's going to allow for formation of this membrane attack complex. What it does is it allows another complement protein called C6 to come next to the membrane. C7 starts to embed itself into the membrane. C8. And then somewhere between 10 to 19 copies of a protein that we call C9 will also embed itself. And the C9 protein embeds itself in a barrel-like structure, so it forms a cylindrical hole or pore through the pathogen membrane or the bacterial cell membrane. So this entire complex, basically, of C6, 7, 8, and 9 associated with C5b is what we call the membrane attack complex, often abbreviated MAC. So once that's formed, you've basically created a pore through the pathogen cell membrane, and that results in loss of membrane integrity, which in the end is going to cause that pathogen to die, or that cell to die. So the classical complement pathway is a very efficient way to lyse the membranes of pathogens, but also to boost the inflammatory response. Because you can see, just in this pathway, we produce 3 different inflammatory mediators, which can help increase blood vessel diameter and result in recruitment of more immune cells to the site of the infection. And we've also have produced a very efficient opsonin, C3b, which is going to help coat the surface of pathogens. And there are actually receptors on phagocytic cells that will recognize this opsonin and enhance the opsonization process, so they will engulf those pathogens and start to destroy them in lysosomes that contain granules or enzymes that work at low pH. So thanks for watching this video. I hope this has helped you understand complement. I have a review sheet for you to work on our Google Drive. You can go to that now and practice this modeling of the classical pathway on your own.